Okay, looking at phospholipids in the cell membrane, you need to be able to describe the structure of the cell membrane, explain the functions of the molecules that make up the cell membrane, and how substances enter and leave cells. The membrane controls the exchange of materials such as nutrients and waste products between the cell and its environment. They enable cells to receive hormone messages, and in chloroplasts they contain the light-absorbing pigments needed for photosynthesis. Taking a look at the structure of membranes, phospholipids help to form the membranes that surround the cells and organelles. Phospholipids are a special type of lipid. One end is soluble in water. This is because one of the 3 fatty acid molecules is replaced by a phosphate group, which is polar. The phosphate group is hydrophilic and makes the head of the phospholipid molecule hydrophilic. The remaining hydrocarbon tails are hydrophobic. The hydrophilic head in the watery solution and then the hydrophobic tails form a layer that is impermeable to the hydrophilic substances. Taking a look at the diagram, this is representing the head again, which is hydrophilic, contains that phosphate group. These are your 2 tails, they're hydrophobic. When phospholipid molecules are spread out over the surface of water, they form a single layer with their heads in the water because the heads are polar, hydrophilic. Their tails project out of the water because they are nonpolar, hydrophobic. If phospholipids are mixed with water, they form either a ball-like structure called micelle or sheet-like structure called bilayers. A micelle is basically a group or cluster of surfactant phospholipid molecules dispersed in a liquid. Taking a look at the picture, when you have water substance present, here you're forming that single layer. These represent the heads, these are the tails. Remember, the head is going to be hydrophilic. You can also form the ball-like structure. These little circles down here are representing the heads, and then the tails are inside, away from the water, because they're hydrophobic. You can also have the bilayer. On the bilayer, again, you've got the heads, which are facing the water surface because they're hydrophilic, and then the tails are inside, hydrophobic. And then on this last one, you could have a bilayer that's forming a membrane-bound compartment, again, where you've got the heads here as well as outside, and then inside you would have the layer of tails. The bilayer membrane is about 7 nanometers wide. It contains proteins embedded within that. When you look at a high magnification, 250,000 magnification for the membrane, it's going to appear as 2 dark lines at the edge of a cell. These are the 2 dark lines. Remember, the membrane also contains proteins. These proteins can be seen in certain electron micrographs, which is what you're seeing in the second image, the proteins that are embedded. The cell membrane is known as a fluid mosaic model. The membrane is described as fluid because both phospholipids and proteins can move about by diffusion. The bilayer has a fluidity that we associate with olive oil. The phospholipid molecules move sideways in their layers, and then the proteins can move like an iceberg in the sea. Some of those proteins will remain fixed. The mosaic is the pattern that's produced from the scattered protein molecules. This is what scientists imagine a membrane would look like. The fluid mosaic model is currently accepted model of the membrane proposed by Singer and Nicholson in 1972. Proteins are free to move in a fluid bilayer of phospholipid molecules. Some phospholipid tails are saturated, while others are unsaturated. Unsaturated meaning they contain a double bond. More unsaturation means there's going to be more fluidity in the membrane. Their tails are bent, so they fit together more loosely. Longer tails are also going to have less fluid membrane. Lower temperatures would also lead to a less fluid membrane. Polar molecules are not able to move through the membrane. The membrane becomes semi-permeable by having protein channels. Each protein channel is a specific channel that allows specific materials to cross the membrane. Going from the outside of the cell inside, you would have sugar molecules, amino acids, and water. Moving from inside the cell outside, we would have salt and ammonia. There are three types of lipids: the phospholipids, cholesterol, and glycolipids. Within the membrane, you also have proteins and glycoproteins. Because the tails of phospholipids are nonpolar, it is difficult for polar molecules, the water-soluble molecules, to pass through the membrane. Examples of these water-soluble molecules, again, are sugars, amino acids, and proteins that cannot go through that membrane without the help of those protein channels. Cholesterol has a hydrophilic head and a hydrophobic tail. The cholesterol is going to fit in between the phospholipids. In animal cells, the amount of cholesterol is about the same amount as phospholipids in cell surface membrane. It's less common in plants and absent in prokaryotes. Cholesterol regulates the fluidity and stabilizes membranes. Without cholesterol, the membrane would break and cells would burst. The hydrophobic regions of cholesterol molecules help to prevent ions or polar molecules from passing through the membrane. This is particularly important in the myelin sheath, which surrounds nerve cells. The myelin sheath is made up of many layers of cell surface membrane. Leakage of ions would slow down nerve impulses. The myelin sheath is acting as an insulator layer around the nerve cells. Taking a look at cholesterol again, here's your heads and the tails of the membrane. Again, the orange ones are representing heads and then the little tails. And then this is representing the cholesterol. Cholesterol is going to fit in between your phospholipids. So this gray one's are cholesterol. Here's the second one, third one, and then a fourth one. They fit between. You also have the protein channels, which we're going to discuss here in just a moment, that also help allow substances to move across the membrane. This is representing a nerve cell, and then you have on here that myelin sheath, which is again acting as an insulator. There's more to the membrane than just the lipids. You do have the lipid bilayer, okay, the two layers, but there's transmembrane proteins, in other words, those integral proteins that are embedded in the phospholipid bilayer. They create semi-permeable channels, so substances are able to pass through. Protein channels in a lipid bilayer, that's what these purple structures are showing. The integral proteins go all the way through, and then you have some peripheral proteins that are going to stay on the surface, and sometimes a peripheral protein will attach to an integral protein. Amino acids are also important. We have different classes of amino acids. As we take a look at the amino acids listed here, these are all example of nonpolar and hydrophobic amino acids. A second look at amino acids for another group. These are representing polar and hydrophilic amino acids. And it's important to know if they're polar or nonpolar to determine how they're going to be able to move across the membrane. Remember that there's proteins that are going to help move substances across the membrane. You have a polar area of a protein and a nonpolar area of the protein. Looking at the membrane, we have nonpolar amino acids, which are going to be hydrophobic. They anchor the protein in the membrane on the outer surface of the membrane in the fluid. The polar amino acids are hydrophilic. They're going to extend into the extracellular fluid and into the cytosol. Cytosol is the aqueous component of the cytoplasm of a cell where you would find various organelles suspended. Looking at many functions of membrane proteins, you have the outside of the cell, the plasma membrane, and then inside the cell. And we have to be able to get substances back and forth. So there's a transporter. It's kind of like a channel where substances can move through the channel. You could have enzyme activity occurring, cell surface receptors, cell surface identity markers, which will identify antigens, cell adhesion, and then the attachment to the cytoskeleton. To know which type of protein is needed, there's a signal transduction, which is going to be transmitting a signal from outside the cell to the cell nucleus. Kind of like receiving a hormone is going to trigger a receptor on the inside of the cell that then signals to the nucleus the type of protein that's going to be needed. Proteins determine the membrane-specific function. Cell membrane and organelle membranes each have a unique collection of proteins. You have peripheral proteins. These are the ones that are going to sit on the surface of the membrane, cell surface identity markers such as antigens. And then the integral proteins, these are the ones that actually go through the membrane. Transmembrane proteins such as a transport protein or the channels or using the permeases, which are pumps to allow substances to move through them. Looking again at the proteins in the membrane, your intrinsic, those are your integral. They're found in the inner layer, so intrinsic, found in the inner layer. They're the integral proteins. Way to kind of help y'all remember. Okay, these are going to be the most common ones. Again, they're going to pass through the membrane. And then your extrinsic, X are the peripheral ones. These are found on the inner or outer surface of the membrane. So they're going to be on the surfaces of them. Sometimes you can even find the peripheral will attach to an integral protein. Here's a peripheral one here. Intrinsic proteins, they stay in the membrane due to its hydrophobic and hydrophilic regions. Most flat-like mobile icebergs, although some are fixed like islands, to structures inside or outside the cell and do not move. So these are representing your intrinsic, the ones that are passing through, to provide a channel for substances to move across the membrane. Extrinsic proteins, Many are bound to the intrinsic proteins, in other words, attached to those integral proteins. Some are held in place by binding to molecules inside or outside the cell. Looking at different roles of proteins in the membranes. Transport proteins that are hydrophilic channels, allowing for ions or polar molecules to be able to move back and forth across the membrane. We have enzymes on plasma membrane on the small intestine surface that's going to hydrolyze disaccharides. Proteins and organelle membranes for photosynthesis and cell respiration. You have transporters, anchors, receptors, and enzymes. Many proteins and lipids in membranes have carbohydrate chains attached to the face of the outside of the membrane. Glycolipids is when you have a carbohydrate attached to a lipid, and glycoprotein is when you have carbohydrates attached to a protein. Glycolipids and glycoproteins form hydrogen bonds with water to stabilize the membrane. They form a sugar coating on the membrane that's called a glycocalyx. It acts as a receptor for molecules. Within the cytoskeleton, there's proteins attached to the cytoskeleton to help maintain and decide the shape of the cell. Carbohydrate chains help glycoprotein and glycolipids act as receptor molecules, which bind with particular substances at the cell's surface membrane. Taking a look at signaling receptors, cell signaling. This is important because it helps explain how living organisms control and coordinate their bodies. Cell signaling, the molecular mechanism by which cells detect and respond to external stimuli, including communication between cells, basically getting information from one place to another. Coordinate activities of animal cells, recognize messenger molecules like hormones, which is a chemical signaling, and neurotransmitters, which is an electrical signaling. When molecules bind with receptors, it triggers a series of chemical reactions within the cell. A stimulus causes the cell to secrete a specific chemical called a ligand. An example would be a glucagon is secreted by cells in the pancreas in response to a drop in blood sugar. The ligand is transported to the target cells. Signaling molecules are usually relatively small and easy for transport. The ligand binds to cell surface receptors on the target cells. The cell surface receptor is a specific shape and recognizes the ligand. Changing the shape of the receptor allows it to interact with the next component. This is called a transduction. That transduction occurs during a cell signaling that is a process of converting a signal from one method of transmission to another. The next component of the signaling pathway is often a G protein, which acts as a switch to bring about the release of a second messenger. The second messenger diffuses through the cell, relaying the message. The stimulation of one receptor molecule results in many second messenger molecules being made in response. This activates an enzyme. The enzymes are produced, which brings about the change in the cell metabolism. G proteins are called this simply because it's GTP, similar to ATP, but GTP is guanine triphosphate, which, remember, ATP was the adenine triphosphate. The sequence of events triggered by the G protein is called a signaling cascade. The diagram is a simplified cell signaling pathway involving a second messenger. The examples, hormones, glucagon, and adrenaline. We will go into this in a later chapter. Other ways a receptor can alter the activity of a cell: the opening of an ion channel resulting in a change in membrane potential, acting directly as a membrane-bound enzyme, or acting as an intracellular receptor when the initial signal passes straight through the cell surface membrane. An example of cell signaling is regulating the insulin in the bloodstream. This is something we will go into in more detail at a later chapter. Another example of cell signaling is endocytosis, binding to molecules that are parts of the structures that are going to be engulfed. You have your ligand. It's going to bond with a receptor. You'll notice how these are going to fit together nicely, and then that's going to allow the cell surface membrane to engulf the structures. Again, we will go into these in greater detail in a later chapter. You can have cell marker receptors such as antigens, allow for cell-to-cell recognition. Each cell type has its own specific antigen, similar to how different countries have different flags. Example of this would be ABO blood group. The antigens are glycolipids and glycoproteins, which have similar differences in their carbohydrate chains.